Hello, everyone. Pam Marshall again, and I wanted to start fielding questions in the chat. There was one thing that I wanted to make sure that all participants were aware of, which is that all of the documents are on the website in the schedule, and it's not just the Common Core outline. In addition to the outline, we also developed lesson plans, activities, rubrics, and handouts that you can look at to see whether or not some of those might help you in being able to organize and develop your own cure. So I don't see any questions in the chat yet. Anyone want to follow up? All right, we have a great question in the chat, which is, do you have any recommendations for modifications when working with first generation students or students with limited scientific backgrounds? So who would like to field that? I can tell you that, in fact, our college, New College, is already identified, our campus is already identified as a Hispanic serving institution, as well as we have a very high percentage of first generation students, I believe over 40%. So that's our student population as well, but we will see what the other team members have to say.
So Pam, if I may, one of the things that really I think was great about our iCure is that we had both mathematical and science majors, and we encountered some of the same kinds of issues in the sense that how do you convince a math major to take a class when they literally can barely tell the difference between a plant and a fungus? So one of the ways we structure the course is that previous content is not necessary, right? There are no tests. There are none of those kinds of learning assessments. They work together as teams. This is also, I think, really effective at reaching those first generation students and other students not generally currently represented well in the sciences. Because this opens them up to being able to ask questions in a supportive environment and not to be supported. And so I think the class in and of itself is very successful at that.
All right, would anyone else like to add? Okay, so we have Jennifer.
Yes. So I currently work at Gateway Community College. I switched over from ASU, and we're also a Hispanic serving institution, and many of my students are first generation. So one of the modifications that I make is to spend more time going over the literature search, how you do it, but also how you read scientific papers. And so I find that investing more time up front helps my students prepare for the research.
Okay, perfect. So we have lots of questions. I thank you very much. The question is, what course or courses might these labs be most appropriate? And also, how do you tie the principles from lectures into the research? Our iCure is a standalone course-based undergraduate research experience, the way that we have it organized, but we do have Dr. Hackney Price, I think, who can talk a little bit about integrating that into a different course. So maybe she could start us off.
Yeah, sure, happy to. So there's different ways to incorporate this into different classes, of course. So I taught a mini cure in which it was a research-based course where most of the students were in the classroom most of the time, but then we had 4 weeks where we brought them into the lab and let them do the actual hands-on portion of the experiments that they worked out. But all of the other stuff was the literature search and all that was done in the classroom. So that was kind of an unusual setting. I also taught this as a full semester course, and I'm actually going to tackle another question that I saw in the chat here. So this was during the pandemic, so just this past semester, and I set up more of a developmental biology cure, just because that's more what my research is. And I had some students that were online and some students that were in the classroom, and that presented its own unique types of challenges. But what I found works really well is utilizing the breakout rooms on Zoom to let the students talk to one another. And if you use that, you could have groups that are both in class and online working together. As long as the in-class folks bring their laptops, then they can collaborate and kind of coordinate their online projects with whatever's working in the laboratory. But I found for the specific class, it really just depends on what the specialty is of the researcher. I think that's the best way to shape it towards a specific class, is to have a specific research topic that's related.
Thank you. I also wanted to highlight that our keynote speaker will be talking some about that later on today. Does anyone else want to answer either one of those questions? The other question was, how do you modify it for a hybrid class or Zoom online, which is not something that I've done.
So I can speak to that. I am in the process of developing a cure for general biology for allied health majors and environmental biology, both of which are synchronous online. But for my general biology, I have students coming in for a lab, and then the rest of the course is online. So I have modified it that way so that we get a little bit of face-to-face time for like the literature search and data analysis, things like that. I've also created a way for students to collaborate with each other online through Google Documents or, using Google Drive.
So here's a follow-up to the first question. How do you tie principles from the lectures into the research? How do you make sure that your student learning objectives for the lab are met when you modify it for a cure?
So Pam, I can try to take that one. In the past, I actually made a mini cure inside of the general biology course, the 182 Bio 182 course. And what that one did is I basically tried to make a cure that would be reflective of the kinds of content issues I wanted to cover. But realize too, in a class like that, any kind of science I'm doing is going to cover a lot of the objectives of the course. The objectives of the course, things like presenting data in a quantitative and a graphical manner. That's always going to be covered in a cure. The cure I used in that course was where I would have students raise butterflies from egg all the way through adulthood. And then they would design an experiment about part of that course to understand something about the life cycle of the caterpillar. So those are some of the ways that you can try to tie in the lab to the course. It's obviously very specific to whatever course you're teaching. If you do a mini cure like my colleague, Dr. Hackney Price, did in her developmental class, then obviously she's going to have much different content needs and would do something much different than what I did. So a lot of that's going to depend on your objectives. But generally speaking, anytime you're having students doing science, they really are meeting what should be some of the objectives of all of our science courses, I think. having them create hypotheses, having them analyze and present the results of that analysis in both graphical and quantitative formats. So I think they're very helpful for those things as well.
I wanted to follow up real quick with that. I mean, many courses do have very specific student learning objectives that you do have to think about being able to be met. But some of the pushback that I've heard and I've got, I've been able to push back a little bit and say, you know what? They don't remember what they do in the lab now. So whether or not we're meeting the learning objectives really doesn't particularly matter, because I can ask sometimes students in upper level classes and say, hey, remember when you did the such and such lab in your first year or your genetics class? No. So I would like to think that one of the learning objectives is they remembered something. Maybe it's not. So we've got a question from Dr. Sandrin, and she asks, what scaffolding do you provide to help students develop a hypothesis? And I believe Dr. Broach would like to cover that. Jen, you're muted, Jen.
Sorry, I apologize. So I know, sorry, of course I was the one that messed it up. Don't worry about it. So Susie, I wanted to address that because we have a whole series of like activities and things to really help them develop their research question, because as Ken had said, they do the literature review. So with Ken's intro, they do the literature review, but oftentimes students don't have a question that's actually testable. So they have these big, broad, huge goals that may or may not be attainable. And so we have a set of activities that we'll discuss, actually it's on the agenda for tomorrow, that we'll discuss that it kind of helps them narrow it down. It helps them choose very specific things that they can actually measure, because it's important that they can actually measure it and test it and actually come up with their conclusion. So that's our opportunity to kind of guide them into making a testable hypothesis. So we actually do take great care in having them think about all of the aspects of their research question, and in a way that will also address one of the other questions, in a way that will start setting them up for very basic statistical analyses, because not all of them have had that class before. So it starts setting them up to doing some very standard analyses. I mean, not all of them will end up that way, but we want to keep it simple, like the KISS method, and then we did talk about keep it simple, stupid. But we want to make sure that they come up with something that they can actually test at the end. So there are a number of activities and a number of worksheets in the documents that help them do that.
Okay, so we had a couple of questions. I think we can kind of wrap them all together. So the question is, when you say mini cure, is that only for a few weeks of lab? So yes, the mini cure is only for a few weeks of lab, as opposed to the entire semester. And another question was for microlab, we have a state mandate to teach certain techniques like gram stain, even if they don't remember it. Yes, there are some states that have that. And so in that case, people have actually taken and modified our Common Course outline and the documents and made things shorter so that part of the class is meeting some of those state mandates that are very specific, like gram stain, and then other parts of the lab cover than more of a research-based or a discovery-based. So that is, I think, the answer to those questions, unless there are additional colleagues who want to answer those.
I'd like to hop in there. So when I'm thinking about incorporating specific techniques, which I did in this previous semester, I try to tie them to the research as much as possible. But for the fly lab that I did, I had a couple weeks where I focused on some core techniques that they would be able to apply to many different research projects. So they got familiar with working with the flies, they learned about fly husbandry and a bit about fly genetics, and then they could move on to actually doing their own experiments. So I try to teach some core techniques first and then move on to the rest, to their own experiments.
So there was a couple of housekeeping questions, unless others want to talk about mini cures. So one of the questions was how much time the students have for the experiments. And in my case, it was weeks. I think in the Common Course outline, we have 4 weeks where the students are actually doing experimentation. And for what I was doing, 4 weeks covered pretty much what they wanted to do. But what I found was that because the different groups were doing different things, and we met once a week for 4 hours, so that's how ours is scheduled and organized, meet once a week for 4 hours, they would need to come in on different days and check different things. And this was a spring, and I believe during spring break, we were in the lab each day in spring break. Different groups were coming in and wanting to do different things. So I would say that one of the differences between perhaps the iCure and if you are placing a cure into an existing course is that the students are opting into our iCure. They're choosing to take the iCure because they either haven't heard, about Dr. Marshall, for example, or they are interested in research, or they've heard that it's a cool experience, or they see the poster that we have that says no tests. And so all of the students that were in my group, my 2 classes, were highly enthusiastic. They wanted to do what they wanted. They wanted to be there, and they wanted to do these things. So there was another question. So then Dr. Mateer asked, how do you ensure that team members are pulling their weight? I didn't need to, because all of my students wanted to be there. I think in other courses where we they maybe don't want to be there, yeah, we do peer reviews, and I have undergraduate TAs circling around and actually paying attention to who's doing what. So so in terms of that, how does everybody else ensure that team members are pulling their weight?
Pam, what I've done in the past, I use a lot of techniques like you do, being very interactive, interacting into the class. But one of the other tools I've used is, instead of a final exam, I do a final survey. And so the students get 100% credit, they fill it out. But one of the questions I'll put on it is, what were your teammates' names? It's always interesting to see if they can remember the last names of teammates. And then I have them evaluate the other teammates. And so they know, everyone knows they're going to be peer evaluated, and I can use that information in order to better assess contributions by group members.
So I want to add to that. So my only issue with having students that weren't really pulling their weight was in an all online group that I had in class this last semester. So there was one who attended the live sessions for every class period, but her 2 partners kind of communicated to me via email, and they were very spotty with communicating with their partner, and it seemed like she was pulling a lot of the weight of the group. So unfortunately, that involved me kind of hounding the other groups and asking, okay, what specifically are you working on? Send me a copy of that you're working on so I know what you did. But I think having peer evaluations would help with that. And also having something similar to author contribution forms, saying, okay, you have to say exactly what you did to contribute to each part of this project, and everybody else in the group has to sign off on that, I think would go a long way in helping out with that.
I love that, author contribution forms. I'm going to steal that. Thank you. So we had another question that was, should teams work together on each task like data analysis, or should members of the team have specific tasks like real science? And I'll answer real quickly. In my case, I let the groups decide, and most of the groups preferred to work together on all of the tasks. And I was surprised that they were not using the divide and conquer like students typically do. But I think, again, it was because they were all so interested in the results, you know. I mean, they came in during spring break. So that was my experience, was that they didn't try to, you know, you write the introduction and you write this. They actually were all working together. So I saw a real ownership in mine. Does anyone have any thoughts on whether the team should work together or they should distribute tasks?
So I have a slightly different point of view because my students are the statistics majors. So they're more of the quantitative people. And I was so adamant about them not just going in and playing the role of data analysis. Like, you guys do the science, I'm going to do the data analysis, and that's how it's going to work. I really wanted them, and that was the whole goal of this project, was to get them to be integrated. And I have to say that, I mean, my students were actually one of the ones that had submitted to the undergraduate research journal. I've had multiple statisticians do that, and that it's amazing to see them dressed up in full hazmat suits, going out into the field and collecting data. And that connection is so much more important than them being able to do the statistical analyses for the group, because then they really integrated themselves. And it was, I mean, not only did they do the cure, they did a second cure, and then they continued on the research just on their own. And to be honest, I didn't really realize that they would actually buy in that much. And I think, and they only bought in because they were involved in every aspect of the research. I mean, as a statistician, sometimes we're just given data often, and we're not really involved in the whole scientific process. So I really feel very passionate about not falling in a role and really being more diverse and really working as a group. So that's just from my point of view. Like, I just know how much my students gained from that experience.
In my first cure, I had 2 statistics majors, and I absolutely told them that you're going to learn how to pipette. You are going to do the, you know, figure out what your hypothesis is and so forth. And so I didn't let them, I did not let the non-statistics majors get away with treating the statistics majors like, you know, they were just going to do the data analysis. And in fact, you know, they came in and they both, you know, they both were like, I don't know how to use a pipetter. I was like, okay, no problem. It'll take, you know, 10 minutes to learn how to use a pipetter at the most. So I think that that's another part that really was very energizing to me, was to show, you know, anybody who wanted to learn how to use a pipetter, how to use a pipetter. We had a quick question, which was, what are the prerequisites, or do we require the students to have statistics before they are in the iCure that we teach? And the prerequisites are Gen Bio for the biology majors, I think a little bit of math if they're computing majors, and what for the statistics majors? Nothing, right? Yeah. So no, we don't expect students to have much statistics at all. And in fact, I'll tell you, one of the instructors doesn't have any statistics either. So anyone else want to talk about what they think that a successful student in iCure looks like?
I'll take that. So, in my opinion, a successful student for the iCure is a student that is enthusiastic about the research, that gains basic skills and an idea of what entering a research field would be like. Now, that's at the end of the cure, obviously. At the beginning of the cure, mostly a successful student is one that is open to learning new skills like statistics or data presentation, things like that. So really a student that's ready to buy into the whole process.
Yeah, I 100% agree. Ken, Jen?
Yes, so I had one student in my classes last semester who got very frustrated when his experiment did not work. And he said, "Oh, I'm going to fail the class. There's no way I'm learning anything." I said, "Hold on, back up. Did you learn how to form a hypothesis?" He said, "Well, yeah." I said, "Okay, did you come up with an experimental protocol?" He said, "Well, yeah." And as I walked him through all of these things he did learn, his whole attitude turned around. So I think a lot of student success is showing them that it's not necessarily about the results, but about, did they learn the process? And if they can take that message home with them, I think that's a success.
Okay, we have just a couple more minutes before we'll go into the next videos, but I think we can answer this question. How much as a coordinator must help and allow students to explore independently? In both cures that I taught, we had a general framework and a general course outline, a general question that the students were going to explore, and then truthfully, they went off in all different directions, and I didn't have to support them very much at all because they were very interested. And so I just allowed them to explore independently, and they did. Anyone have a different experience?
Yeah, so for my semester-long cure, it was the same thing as what Pam was describing, that I let the students have more free reign in deciding what they wanted to study and how they wanted to approach it. But for my 4-week-long mini cure, that's where I really kind of reined them in and said, these are the variables that you get to test. And in this case, they were testing the effects of different food supplements on fly fertility. So they got to pick whatever food, whatever additive they wanted to include in the food, but that was the variable that they were testing. So it really depended on how much time we had as far as how much free reign I let the students have.
All right, we've got maybe one more minute, and there's one more question in the chat that I wanted to bring up, which was, I want to try to incorporate some of these cures as a required research methods class for CJ majors. I think that's criminal justice. And the question was, any suggestions? I think that is a great question to explore this afternoon when we get into the Zoom, if you can make it, because I think that that is going to be a little bit more than we probably can tackle right now in the next 30 seconds. So hopefully you can make it to the Zoom. Did everybody receive the email with the Zoom link? And it's long and it has a link and it has the phone numbers and everything. Okay. So I just wanted to thank everybody for this great chat, and next up, we will have Ken Sweat with his section about laboratory techniques for phytoremediation. This was a cure that he developed with students, and he's going to walk you through a little bit about how to do this cure if you would be interested in a cure on phytoremediation.